In this video, we're going to talk about what are proteins. In particular, we're going to talk about how the body makes a protein, what are the different levels of protein structure, which are primary, secondary, and tertiary. What is the protein data bank, and how can we quantify a protein structure using graph theory? So first, what are proteins? Proteins are large molecules that our body uses to perform most of the functions. For example, there are proteins that are in the cell's membranes, and their function is to move things in and out of the cell. Here the keyword is function. When we think of our body operating, carrying on all the biological processes that it does, there's always a protein carrying on that function. How are proteins found in our body? Well, our body makes proteins. How does it make a protein? The information or instructions to make a protein are encoded in our DNA. Those sets of instructions to make a protein are what we commonly call genes. So our DNA sequence, the entire sequence, is not completely a set of genes. Rather, we have these small subsequences. And these small subsequences are the genes. And the genes encode the proteins. So this is the basic paradigm in biology. That you have a gene. And when our body needs a protein, that gene is transcribed, which means it's copied into another strand, which is RNA. And the RNA strand is identical to one of the strands in DNA, except that the T is changed to U. And then once we have a transcription of the gene, that encodes the protein. Every block of 3 represents a unique protein. Well, actually a unique amino acid. And then the sequence of amino acids is the protein. There are 20 distinct amino acids that our body uses to construct a protein. The 20 letters represent the 20 different amino acids. And so we can think of the beginning of a protein structure as simply a sequence of amino acids. So for example, the sequence below has the amino acids M, D, F, R, and so forth. And this sequence of amino acids is called the primary structure of a protein. Now, what are these amino acids and what do they look like? All 20 amino acids have the same chemical structure, except for the part that's represented here by the box labeled R. So how they differ is what's in the box labeled R. But the part where they are the same is called the backbone of the amino acid. So here we see we have nitrogen, 2 carbons, and 2 oxygens, together with some attached hydrogens that form the backbone of the amino acid. The carbon that the box R is attached to is called the central carbon. So since there are 20 different amino acids, there are 20 different things that go in the box labeled R. So for example, on the upper right, where we have a central carbon being pointed to, so in these schematic drawings, we're saying at the top, this is where it attaches to the central carbon. And then what's in the R is a different chemical structure for each of the 20 amino acids. For N, up there in the top right, we have a carbon attached to a carbon, which in turn has an oxygen and a nitrogen attached to it. Now the smallest one is in the bottom left. We have C and A. That's cysteine for C. So let's go back and recall what we said. They all have the same backbone. But in the box that's labeled R, we have a different chemical structure. So amino acid C is small. In its box, in its residue box, as the R stands for residue, is a carbon atom, and attached to that carbon atom is a sulfur atom, and that's the amino acid cysteine. Another way to visualize the primary protein structure rather than a sequence of letters is a sequence then of these amino acids, looking like a string of pearls. In each of these circles, each of these pearls represents a unique amino acid. So if we zoom in on the first 4, we see we have phenylalanine, leucine, serine, and cysteine. We could zoom in on each of those to see its chemical structure. So that was the primary structure of a protein. You have a sequence of amino acids. But now, these amino acids, some repel each other, some are drawn together. And so once this sequence is constructed, it begins to fold upon itself, forming a 3-dimensional molecule. You have substructures, and the substructures are called the secondary. And then the entire molecule is called a tertiary. So in the protein on the right, the green twisty parts give an example of secondary structure that's called an alpha helix. We have, it looks like 3 green alpha helices. We also have, up on the top, the part that's blue, that substructure is called a beta strand, and also a blue alpha helix. And then, as I mentioned, the entire structure, once it's a 3-dimensional structure like this, is called a tertiary structure of the protein. I've circled where the amino acid sequence, its primary structure begins and then ends. Now, an interesting problem in computational biology is to discover what is the tertiary structure of a protein. How could we not know? Well, it turns out that it's fairly easy, relatively speaking, to identify a gene in your DNA sequence. So if we identify a gene, the assumption is that it encodes a protein, but we may not know what the protein is and what its function is. So a big problem in computational biology is, given the primary structure of a protein, can we predict its 3D structure? And then, given its 3D structure, does that inform us about its function? So there are a lot of proteins that we do not know what the 3D structure is. On the other hand, the protein data bank is growing at an amazing rate because of the improved technology that allows researchers to determine the protein's 3D structure. Once a protein's 3D structure has been determined, that information is deposited in the protein data bank. What we're going to do with this lesson is utilize a web server that will read the information about the 3D structure from the protein data bank. You tell it what protein, it will go to the protein data bank and get the information about the 3D coordinates of every amino acid in that protein, and then it will construct a graph. Each vertex represents an amino acid, and two vertices are connected if those two amino acids are within proximity in the 3D structure. Usually the cutoff for proximity is 7 angstroms. So in our homework, we'll go and investigate what a graph looks like that represents a protein structure. So we have given a brief primer of what proteins are and how a gene encodes a protein. What are the levels of protein structure, the primary, the secondary, and tertiary? And what is the protein data bank? It's a repository of all known three-dimensional structures of protein.